Hello everyone, my name is Insop Choi, working in JNU-US lab at Icahn School of Medicine at Mount Sinai in New York City. First of all, thanks to the BRIC team for inviting me and organizing this session from several months ago. And also personally, it's great pleasure to share my research here for the first time in BRIC seminar. Today, my topic is the role of microglial autophagy in Parkinson's disease. So I will give an introduction one by one. Parkinson's disease is the second most common neurodegenerative disorder. Hallmark of PD is the loss of the dopaminergic neuron as substantia nigra pars compactor. So as you can see here, PD postmortem tissue, you can see the loss of pigmented dopaminergic neuron. Also, you can see the dopaminergic neuron marker TH loss in the PD postmortem brain. Since this area is important for the movement control, so PD has accompanied motor-related symptoms such as tremor, bradykinesia, rigidity, and gait and balance problem. Before and after motor-related symptoms, there are also non-motor-related symptoms such as constipation, loss of smell, mood disorder, and also cognitive problem at the end stage of PD. Histological studies showed the formation of a protein aggregate called the Lewy body. And then later people found out the alpha-synuclein protein is the major component of the Lewy body. And what's the relationship between alpha-synuclein and Parkinson's disease? Late 1990 or early 2000, 3 different point mutations were found in alpha-synuclein locus reported in the familiar PD case. And then Anna Maria Krubor, David Schultz's lab, they found out the mutant alpha-synuclein become resistant to degradation system called CMA. And early 2000, there's also duplication and triplication mutation were found in a familiar PD case. And GWAS study with the on PD brain has consistently shown the alpha-synuclein as a top risk factor for Parkinson's disease. And Janice Rudolph group, MIT group, they tested one of a risk SNP of alpha-synuclein, and they found out it really caused the increase of alpha-synuclein, even though it's mild increase. So overall, I can say the amount of alpha-synuclein is directly linked to the PD. In early 2000, Brock's group at Germany, they found out the protein aggregate, Lewy body synuclein aggregate, can spread from subcortical to the cortical area as disease progress, and also is correlated with the symptomatic stage. Another evidence can be found in a postmortem study of a Parkinson's disease patient who received a mesencephalic embryonic tissue for therapeutic purpose. In their postmortem study, people found out a synuclein aggregate in the host tissue, which is expected, or also in a transplanted tissue, which might indicate the alpha-synuclein aggregate are propagated from the host to the transplanted tissue. So after this finding, the people has been interested in the secretion feature of alpha-synuclein. In human, alpha-synuclein exists in a plasma and CSF and brain's interstitial fluid. And secretion of alpha-synuclein is known to be controlled by neuroactivity. And secretion of alpha-synuclein occurs in cortical primary neuron, and secretion of alpha-synuclein can be increased in under stressful conditions such as mitochondrial, proteasomal, or lysosomal, and also oxidative stress condition in neuron or neuronal cell line. Overall, this observation might indicate the uptake and clearance of extracellular alpha-synuclein might be important to prevent the propagation of alpha-synuclein aggregate and also disease progression. What about microglia? So as you know, in our body, there are tissue-specific macrophage, and mostly derived from the hematopoietic stem cell lineage. But microglia, which is the brain macrophage, is derived from the yolk sac precursor. So precursor infiltrate into the brain and proliferate and differentiate into the major microglia. In homeostatic condition, especially during the developmental stage, microglia is known to regulate the synapse formation, including synapse pruning process. And also it can eat up the apoptotic neuron generated from the developmental stage, also involved in the learning and memory process. In a pathological condition, microglia can excessively eat up the synapse, which can be detrimental for neuronal viability. And also microglia are known to regulate the amyloid plaque formation. Also, it can degrade the amyloid monomer or oligomer. So I had a simple hypothesis that microglia keep alpha-synuclein homeostasis by clearance. Alpha-synuclein can be secreted out from the normal neuron, or it can be passively leaked from the dying neuron and exposed to the neighboring cell, such as neuron, microglia, and astrocyte. And in this study, I focus on the microglia. To test this hypothesis, I adopted two different Parkinson's disease animal models that express the wild-type human alpha-synuclein in neuron-specific manner. First model, I adopted the AAV alpha-synuclein model. So we got an AAV virus that have a gene encoding either GFP or wild-type human alpha-synuclein under synapsin promoter, which is neuron-specific. After injecting AAV GFP virus into substantial Niagara area, I confirmed the GFP signal are colocalized with the neuronal marker NUN and dopaminergic neuronal marker TH, but it's not colocalized with the astrocyte marker GFAP or alpha-4, also not colocalized with the IB1. So this confirmed the specificity of the neuron-specific expression system. Consistent with the other report, AAV-induced alpha-synuclein overexpression caused a massive dopamine neuron degeneration at the east-retinal side compared to the contralateral side. Also, I collect the whole brain lysate from at the 2 weeks or 4 weeks, 10 weeks after AAV injection, and I can see the dopamine transporter, either TH level, are gradually decreased as disease progressed. Another model, we adopt the alpha-synuclein transgene line that express, again, wild-type human alpha-synuclein under Taiwan promoter, also known to neuron-specific. This model is developed by Michael J. Fox Foundation, and kindly they provide dopamine neuron counting data with using 1-year-old mice. We can see there's no obvious dopamine neuron degeneration, probably due to the mild expression compared to the AAV. So I asked how microglia react in two different PD models. First of all, I analyzed the microglia morphology because it is a very sensitive indicator of a status of a microglia. So after injecting AAV alpha-synuclein, either GFP at the substantial Niagara, and at the 6th week after injection, I look at the microglia at the striatum. So as you can see here, the filament length becomes shortened, and the number of filament terminal points also reduced, indicative of compromised microglia morphology. Whereas I can see the microglia cell number significantly increase. So this is the typical marker of microgliosis, microglia activation. And also I applied the disease-associated microglia gene set, originally established in Alzheimer's disease model. And I can see that some of the disease-associated signature genes are also regulated in the microglia in this AAV PD model. Importantly, I wanted to ask whether microglia can take up the alpha-synuclein released from the neuron. For this, I directly collect the microglia by flow cytometry at 2 weeks, 4 weeks, and 10 weeks after AAV injection. And then I can see the clear human alpha-synuclein signal in microglia lysate. Also, I can see at the striatum, I can visualize the microglia containing the human alpha-synuclein inside of them. In another transgenic model, I analyzed the microglia morphology at the 10 months old against a striatum. I can see the similar phenotype, shortening of filament length, and also reduce the number of filament terminal point, also a meaningful increase of a microglia number. So again, this indicates the microglia activation. The same way, I collect the microglia by flow cytometry at the 7 weeks or 3 months or 1-year-old mice. And as you can see here, I confirm the alpha-synuclein signal in the microglia lysate. Also, at the same time, I can visualize the microglia containing the human alpha-synuclein in their body. So next, I asked how microglia will handle this alpha-synuclein after takeoff. My PI genuine US lab has investigated the role of autophagy in a neurodegeneration. Autophagy is the bulk degradation pathway responsible for the clearance of protein aggregates and damage to cellular organelle. You can see PHAGO4, which is the initial component of autophagosome. They can recognize its target through the selective autophagy receptor. P62 is the very well-known selective autophagy receptor. P62 can interact with the lipid LC3, which is the basic component of PHAGO4 through Lial motif. And also it can interact with the ubiquitinated target through the UBA domain. After recognition, PHAGO4 become elongated and then make autophagosome to surround its target and then fuse with the lysosome for the degradation. It's known the removal of autophagy essential genes in neuron cause the neurodegeneration. The role of autophagy microglia is still unclear. So if I look at the LC3 and ubiquitin puncta and P62, I ask whether microglia autophagy machinery can recognize the alpha-synuclein from the neuron. And also, by using ATG14, either ATG7, I ask the impact of autophagy in alpha-synuclein degradation. For the first question, I inject the AAV alpha-synuclein into GFPSH3 mice, which is the autophagosome reporter line. And again, at the striatum, I can see the GFPSH3 puncta are interacting with the human alpha-synuclein inside of the microglia. Also, I can see the similar interaction between alpha-synuclein P62 and also ubiquitin and alpha-synuclein. In addition, I use the cultured primary microglia and I treat the recombinant human alpha-synuclein protein. I confirm the similar interaction between LC3 and synuclein, P62 and synuclein, and ubiquitin puncta and synuclein. Furthermore, I did an immunoprecipitation assay using brain injected with the AAV alpha-synuclein, either from the human transgenic synuclein transgenic line or using cultured microglia treated with the human alpha-synuclein protein. After pull down with the alpha-synuclein, I can confirm the high molecular weight of P62 was interacting with the internalized with the alpha-synuclein, human alpha-synuclein. This was the consistent with the known notion that P62 recognizes ubiquitin target and then become aggregate or it become oligomerized. So it make a kind of a small puncta that can be degraded through the autophagy pathway. So we interpret the high molecular weight of P62 might indicate the oligomerized form of a P62. Then after recognition, what about the degradation? In cultured microglia, when I treat the human alpha-synuclein, as I show you, some of the alpha-synuclein signal was colocalized with the ubiquitin puncta. But also there's a synuclein signal that not overlap with the ubiquitin. But interestingly, at the 24-hour, synuclein ubiquitin double positive puncta disappear. So I used this time frame to see the impact of the autophagy. For this, I cultured the microglia from HG7 conditional knockout using microglia creline. Also, from the culture from the HG14 conditional knockout, I confirmed the HG7 depletion or HG14 depletion, and I observed the accumulation of P62. So far, I introduced the P62 as a selective autophagy receptor, but interestingly, P62 itself is the autophagy substrate. So if autophagy processes are compromised, then P62 started to accumulate. And furthermore, I confirmed the LC3 2-type, lipid LC3 2-type are reduced. So by using this system, I treat the human alpha-synuclein here. And I can see the alpha-synuclein ubiquitin double positive puncta degradation was blocked by in the absence of ATG7, also in the absence of ATG14. To test, to see this phenomenon in the in vivo, now I apply the more specific microglia line, which is tamoxifen inducible. I inject the tamoxifen to induce the Cre recombinase, and then I inject the AAV alpha-synuclein. So as I introduce the P62, I can see the P62 puncta accumulation because compromised autophagy. But interestingly, I can see the P62 and alpha-synuclein double positive puncta also accumulated. In different animal PD model, now I combine the microglia-specific ATG7 deficient mice with the synuclein transgenic line to make a compound mice, and then I inject the tamoxifen and then analyze later. Similar to the AAV model, I can see the accumulation of P62 puncta and also P62 alpha-synuclein double positive puncta. Since it seems that the P62 is an important selective autophagy receptor for recognizing and then degrading the alpha-synuclein through selective autophagy pathway, I look at closely how P62 react in response to the alpha-synuclein for the mechanism study. For this cultured microglia, I treated the human alpha-synuclein protein, and I can see the clear increase of a P62 protein. Also, I can see the transient increase of a P62 mRNA level. To test the contribution of this mRNA increase to the protein, I pre-treat the actinomycin D before treating the alpha-synuclein. As you can see here, the P62 increase by alpha-synuclein protein was blocked by actinomycin D pre-treatment. Accordingly, I observed the alpha-synuclein ubiquitin double positive puncta formation was compromised in the presence of actinomycin D. And I also checked the P62 increase in two different PD model in microglia. You can see the AAV injected AAV alpha-synuclein model. I can see the more P62 puncta at the microglia, and also it happened the same to happen to transgenic line. Even though I didn't show it here, I confirmed the mRNA level increase of P62 after collect the microglia by flow cytometry. To confirm again the role of P62 as a selective autophagy receptor, I cultured the microglia in the P60 knockout background, and I treat alpha-synuclein here. As I show you, alpha-synuclein signal or some of the alpha-synuclein signal was overlapped with the ubiquitin puncta. Interestingly, the synuclein ubiquitin double positive puncta disappear in the absence of P62, which indicate the P62 is required for scavenging the alpha-synuclein and then make a small puncta that can be degraded through the autophagy. The first summary is the wild type alpha-synuclein internalized, and then it triggered the P62 mRNA level and the protein level increase. And then as a selective autophagy receptor, P62 will intermingle with alpha-synuclein, and autophagy machinery can recognize it and then make autophagosome and degrade it through the autophagy pathway. Next, I was curious what mediated alpha-synuclein induced the P62 mRNA increase. Among many different candidate receptors for alpha-synuclein, I can easily narrow down either TR4 or either TR2 due to many publications. TR4 was required for alpha-synuclein induced microglia activation, either astrocyte activation. Even TLR4 was associated with alpha-synuclein clearance and also survival of a dopaminergic neuron. In TLR2 case, the neuron released oligomeric alpha-synuclein known to interact with the TLR2. But interestingly, TLR2 was also associated with the neuron side. TLR2 expression is known to be increased in a neuron at the PD brain, also antagonizing the neuronal TLR to prevent the synucleinopathy. The test is I cultured the microglia again from TLR4 knockout background, and I treat the alpha-synuclein. As you can see here, the alpha-synuclein increased the P62 protein nicely in wild type, but induction was compromised in the absence of TLR4. Also, P62 mRNA level increase was reduced. Also, the alpha-synuclein ubiquitin double positive puncture formation was compromised in the TLR4 knockout background. And I treat the TLR4 specific inhibitor called the TAC242. I pre-treat the TAC242, and then I can see the alpha-synuclein P62 induction by alpha-synuclein was blocked by TAC242. Also consistent with the TLR4 knockout microglia, I can see the alpha-synuclein ubiquitin double positive puncture formation was reduced by TAC242. Importantly, I also confirmed the P62 increased in an AV model in the microglia was reduced in the absence of TLR4. But interestingly, when I look at when I set the different gene that can be activated by as a downstream of a TLR series, like IL-1B or TNF, I can see the induction level was much compromised in TLR4 knockout. But when I compare with the non-treated, still I can see the increase of IL-1B and then TNF alpha. So I speculate this might involve of TLR2. So I pre-treat the TLR2 blocking antibody, clone T2.5. Pre-treating the TLR2 blocking antibody completely suppressed the I1B increase and TNF alpha increase in a TLR4 knockout background. Furthermore, TLR2 blocking antibody inhibitory effect was much stronger at the TLR4. So these data indicate in the absence of TLR4, alpha-synuclein starts to interact with the TLR2. To test this in another system, we reconstitute the TLR4 complex in a HEK cell because HEK cell doesn't express any TLR. And also we co-transfect the NF-kappa B activity reporter, which expressed the luciferase gene under NF-kappa B binding element. Same with the cultural microglia, alpha-synuclein treatment increased NF-kappa B activity. Also, we test the TLR2 homodimer, either TLR1-26 heterodimer, and again, TLR4 complex system, also TLR5. So all known to be expressed at the plasma membrane. So you can see the alpha-synuclein increased the old TLR2 system and TLR4 system, except the TLR5 system, TLR5 signaling. So using this system, I test the preference of alpha-synuclein again. For this, we transfect all together with TLR2 and the TLR4 complex. We treat the LPS, which is TLR4 agonist, increase the NF-kappa B activity, but it's not blocked by TLR2 blocking antibody. But when we treat the PAM3CSK, which is TLR2 agonist, was blocked by TLR2 blocking antibody. So these two validate this specificity of this system. But NF-kappa B activity increased by alpha-synuclein was not affected by TLR2. So based on this observation, we interpret this as alpha-synuclein act on the TLR4 over TLR2, and then turn on NF-kappa B pathway, and then turn on the P62, and then P62 mediate alpha-synuclein degradation through selective autophagy pathway. And we asked what's the final consequence of a microglia-specific autophagy deficient brain in a PD context. For this, I collect the brain that inject with the AAV from the AAV model, either alpha-synuclein transgenic model, and then I did a fractionation assay. Interestingly, I can see the alpha-synuclein, human alpha-synuclein signal at the detergent insoluble fraction was significantly increased. And also in this transgenic line compound mice, I can see the insoluble fraction of alpha-synuclein was meaningfully increased. So this indicates there's accumulation of aggregate alpha-synuclein or misfolded alpha-synuclein in the absence of a microglial autophagy. For the final, we access the dopamine neuron degeneration. So as I introduced earlier, the synuclein transgenic line itself doesn't have an obvious neurodegeneration up to 1-year-old mice. Also, just deleting the HG7 from microglia specifically doesn't cause any dopamine neuron degeneration. This is again 1-year-old. Interestingly, when we combine the microglia specific HG7 mice with the synuclein transgenic line that mildly express the human alpha-synuclein, I can start to see the reduction of the dopamine neuron system, which indicates that there's a synergistic effect of a microglial autophagy deficiency and alpha-synuclein transgenic line. So through this, I can confirm the autophagy deficient microglia cause the accumulation, disrupt the alpha-synuclein homeostasis, and it might lead to the dopamine neuron loss. So this is pretty much it. So we named this microglia specific alpha-synuclein degradation pathway by autophagy as a synuclein phage. But as I show you, the alpha-synuclein can be released from the neuron, and then it activates the microglia. But at the same time, alpha-synuclein triggered the TRR4 signaling to turn on the P62 mRNA level. P62 as a selective autophagy receptor intermingle with the internalized alpha-synuclein from the neuron, and then autophagosome and degrade through the lysosome. So we published this data in the first paper, and then we wrote a small essay about this synuclein phage at the autophagy. So this is pretty much it. And I would like to thank my PI, Jenny Yue. He is a great supporter for me. And also thanks to Steven Sigobin, he performed the mostly luciferase assay. And he took a master degree in the Genius lab. And also thanks to Yunzi Zhang, he injected a lot of AAV virus for me. Also thanks to GIE, who provided AAV vector. And also thanks to Michael Jacks Foundation, who developed this model and also provided dopamine neuron counting data. Actually, we are looking for the postdoc. So whoever interested in the microglia, neurodegeneration, and autophagy is more than welcome to apply. You can directly send an email to my PI, or you can have a question to me. Also, I really thanks to my thesis PI, Jo, at School of Medicine at University. She provided me great training and also great insight about the neuroprotective role of a glial cell, including microglia. So it's great to have some subject that I can pursue throughout my career. Thanks for listening, and I'm happy to answer any question.